Good day, my name is Jakob Nissen. Today I'm presenting my work, Deep Learning for Binning and High-Resolution Taxonomic Profiling of Microbial Genomes. So, why do we want to bin metagenomic sequences? Well, suppose you have a sample containing multiple genomes, and you do next-generation sequencing and obtain reads from your genomes. If you want to find out which species are in your original sample, one common approach is to align your reads to a database of some sort. You may then look at the top hits and determine what's in your sample. But this will obviously not be able to find new species since they're not in the database. Furthermore, even if your species is in the database, any new genomic features like virulence genes are probably not. An alternative is to use de novo assembly. Here your reads are assembled into longer contexts. But especially with metagenomic data, it's normally not possible to assemble one genome into a single context. Instead, your genomes are spread across multiple contexts. In order to obtain your original genomes, you then have to resort to the process of binning. This process is grouping your contexts based on the genome of origin. A common approach to binning is to first take your sequence k-meres and also your sequence depth across the samples and combine it in a statistical model. This model gives the probability that two different contexts belong to the same genome. Using these probabilities, you can then cluster your contexts and obtain your original genomes. But integrating k-meres and depths in a single model is very hard, and it's even harder if you have to make it run in a reasonable amount of time, say hours or days. Our main contribution is to use deep learning instead of an explicit model to automatically integrate these data. We do this with our program called Vamb. In this figure, on the y-axis, we have the number of high-quality genomes reconstructed, and the 4 different bars represent 4 different binners. Our binner is in blue. You can see that it reconstructs more genome than the other 3 binners, MetaBat2, MaxBin, and Canopy. This is true across all our 6 benchmark datasets. The Vamb program works in 3 steps. In the first step, you calculate the k-meres and the depth from the input files, and you represent these data as matrices. In the second step, you transform the matrices using a variational autoencoder, which I will explain in a bit. In the third step, you cluster this transformed data using a novel clustering algorithm to obtain your original genomes. Why do we use a variational autoencoder? Variational autoencoder are Bayesian models that explain how data is generated. It assumes that data is sampled from distributions. In our case, we say that the contexts are sampled from the different genomes. Variational autoencoders also have a prior belief that each distribution looks like the average of the dataset, or at least close to the average. We have this prior here so that we don't infer a bunch of very different genomes, such that not every single context has its own genome. We begin with our context represented as k-meres and depths. In the first step, the variational autoencoder infers a genome from this context. Here, the prior probability can be calculated. It calculates if the inferred genome looks close to the average of the whole dataset. In the second step, a single context is sampled from the genome. Here we can calculate the likelihood. Does the sampled context look like the genome it originated from? When we train our variational autoencoder, we train it to optimize the posterior. That's the product of the prior and the likelihood. In this way, we infer genomes that actually look like our context, and we don't infer too many too specific genomes. A smaller contribution of our work is the process of bin splitting. Normally, when you bin across multiple samples, you're faced with a dilemma. Do you bin your samples together? In that case, you lose information on the relationship between genomes and samples. Furthermore, samples may contaminate each other. Alternatively, you may bin your samples independently. but then you lose depth information across all your different samples. Furthermore, your clusters will be lower density because you have fewer contexts, and this makes them harder to cluster accurately. Our simple approach is first to cluster with all the samples and then split the samples apart. Then you get the best of both worlds. Also, the splitting process reveals a relatedness between bins that were split from the same cluster. After all, if two contexts from different samples were clustered together, they're probably quite similar. This is useful if you want very precise bins. For example, for one of our benchmark datasets, we obtained 6 different clusters that matched to the bacterium Akkermansia municiphala when we blasted it to NCBI's NT database. We then constructed a phylogenetic tree from a core genome alignment of these bins. All 6 clusters were monophyletic according to this tree. Furthermore, the tree was consistent with the blast nucleotide identity levels. This implies that Vamb was able to distinguish the 6 genomes better than blast to NCBI could, since they all blasted to the same genome. The splitting process in Vamb also revealed which genomes across the different samples were similar. And here we obtained a total of 89 high-quality sample-specific genomes from 6 clusters. We built Vamb to scale with the big datasets that are common in metagenomics today. And to stress test it, we tested it on the benchmark dataset released by Almeida et al. In Nature last year. Their dataset contains 1,000 human feces samples and 5.9 million contexts. We binned it with Vamb using a single GPU and spent only 29 GB of RAM and 9 hours. According to the program checkm, we created 4,920 high-quality genomes, compared to the 3,467 that Almeida got using MetaBat 2. That's 41% more. Vamb is free and open software. You can get it on our GitHub website. This work is sponsored by the Novo Nordisk Foundation, Yorks Foundation, the Technical University of Denmark, and the Novo Nordisk Foundation Center for Protein Research. Thank you all very much for listening.